Good morning. Welcome to zoology class. My name is B. Kalpana, Assistant Professor of Zoology in Bingley Government Degree College. Today's the topic is animal cloning. Okay, we already learned about the DNA cloning or gene cloning and rDNA technology. Okay, now today the animal cloning. What is cloning? Cloning means production of same phenotype or same genotype of identical copy of an individual or a cell or a organ or DNA. If DNA is produced, that is DNA cloning. Okay, gene cloning. If a gene is produced, that is gene cloning. If an animal is produced, then it is animal cloning. Okay, for example, natural clones are identical twins. Okay, in which two embryos becomes two halves and develops into new single embryo becomes two halves and develops into two organisms. In the same way, the cloning okay, sorry for the disturbance. Same identical copies, production of identical copies of an individual is called cloning. Okay, next, it can be of three types, that is I already told you, gene cloning or DNA cloning, that is duplication of a string or part of a DNA containing genes in a host bacterium through recombinant DNA technology. Okay, so rDNA technology, what it is? It produces number of copies of a gene, or which gene we want, whatever gene we want, that copy is produced. In the same way, DNA, whatever we want, that DNA part or gene part is produced, is called DNA cloning. Molecular cloning, that is copies of cells made copies of cells are made, resulting in cell line. Next, embryo cloning, that is embryo which is already formed sexually split into two identical halves, that is embryo cloning. So, history of cloning, we heard, many of us, about the dolly, the species of sheep. The Greek word clone refers to the process whereby new plant created from a twin. Hans Driesch separated blastomeres of two-cell embryo of a sea urchin. Hans Spemann repeated in a vertebrate salamander using hair from his baby boy to separate the cells. Okay, cloning by somatic cell nuclear transfer, that is transferring the nucleus of a somatic cell into an oocyte of the same species. World's first cloned mammal from the adult somatic cell is Dolly in 1996 by Wilmot. In cattle, mice, pigs, goats, rabbits by Edward in 2001. So these are the two animals with black faced sheep and white faced sheep. So he has selected these two sheeps. Okay, then types of cloning: recombinant DNA or DNA cloning or gene cloning. Next is reproductive cloning. Next is therapeutic cloning or embryo cloning. Steps in cloning procedure: collection of ovaries or oocytes from the organism, then in vitro maturation of these oocytes, then enucleation of oocytes, that is removal of nucleus from the oocytes, then fusion of somatic cell nucleus with this recipient oocyte. Next, activation and culture of the embryo transfer into recipient animal. This is the procedure in this. See, egg donor is a black faced sheep, somatic cell donor is white faced sheep. Okay, from the egg, after developing this egg, from the egg nucleus is removed and the somatic cell of another sheep has a nucleus. From this somatic cell the nucleus is removed and this nucleus introduced into the recipient ovum cytoplasm. Then it is allowed to develop into an embryo. Then blastocyst is formed and new animals are developed. Okay, these new animals are like white faced sheep. Okay, same diagram in color, that's it, nothing much difference. See the process: induced division of for cloning. Stage 1, cells collected from sheep's bladder. Okay, next nucleus is removed from unfertilized egg of second sheep. Then udder cell is inserted into the egg with no nucleus. Next, insertion is successful. This nucleus enters into the cytoplasm with the electric charge supply. Then next stage, cell begins to divide. So what are the applications of cloning? To produce desired individual for research purpose, to produce desired individual to rapid propagate desired individual, to produce genetic targeted genes or different genes, clones of endangered species. Cloned animal is for testing new drugs and treatment. Drawbacks of clone: very expensive, time consuming, cell mutation may occur, organ rejection because of cell mutation, clones do not always look identical, some clones cannot survive. So this is whole animal cloning. I think you people have understood very well of DNA cloning earlier classes, RDNA technology, and now whole animal cloning is also very easy. I think you people have understood. Next, transgenic animals, next topic. Okay, what is a transgenic animal? A transgenic animal is one that carries a foreign gene has been deliberately inserted into its genome. Its genome of the animal to alter its DNA. This is done to improve the genetic traits of the targeted animals. Transgenesis is the process by which mixing up of genes takes place. Foreign genes are inserted into the germline of the animals, so it can be transmitted to the progeny. Transgenic technology has led to the development of fishes, livestock, and other animals with altered genetic profiles, which are useful to mankind. Initially, for the improvement of genetic traits, what they used to do is selective breeding methods. That is, animals with desired genetic characters were mated to produce an individual with improved genetic characters. Since this technique is time consuming and expensive, it was replaced by rDNA technology, that is transgenic animals. First transgenic animal was a super mouse created by Ralph Brinsters, Pennsylvanian, and Richard Parmiter in 1982. It was created by inserting a human growth hormone gene in mouse genome. The offspring was much larger than the parents. Mouse. Common transgenic experiment. Other animals includes pig, goat, cow, sheep, fish, etc. Next, production of transgenic animals. The methodology. Step 1, construction of a transgene. It mainly consists of three parts. Promoter region, gene to be expressed, and terminator sequence. Next, step 2, introduction of this foreign gene into the animal in two methods: pronuclear microinjection method, embryonic stem cell method. Microinjection method first. A female animal is superovulated and eggs are collected. Superovulated means taken out. Eggs are taken out of that animal, and these eggs are fertilized in vitro, means outside the animal in a laboratory. The transgene contained solution is injected into this male pronucleus using micropipette. See here what happened. Eggs are fertilized. Fertilized means male and female nucleus are present in this ovum. In this larger one is male pronucleus, and to this nucleus we are injecting what is this gene, desired gene. Then eggs with transgenes are kept overnight in an incubator to develop to a 2-cell stage. Then these eggs are implanted into the uterus of pseudo pregnant female, that is female which has been mated with a vasectomized male in the previous night. Next, embryonic stem cell method. Transgenic animals can be created by manipulating embryonic stem cells. Embryonic stem cells are obtained from the inner cell mass of blastocyst. Transgene is incorporated into the embryonic stem cell by microinjection or retrovirus or electroporation. Then these transgenic stem cells are grown in vitro. Then they are inserted into a blastocyst and implanted into a host uterus to grow normally. Okay, this is blastocyst microinjection diagram. This is central one is blastocyst. Okay. Next, step 3, screening for transgenic positives. Transgenic progenies are screened by PCR to examine the site of incorporation of the gene. Some transgenes may not be expressed if integrated into a transcriptionally inactive site. Next, further animal breeding is done to obtain maximal expression. Heterozygous offsprings are mated to form homozygous strains. See, method 1. Method 2. DNA embryonic stem cells selector for cells expressing desired gene. Inject transformed this embryonic stem cells into inner cell mass, that is blastocyst. Pronuclei, that is fertilized egg, desired gene introduced into the male nucleus. Then after developing a 2-cell stage, this embryo is inserted into a female rat. Then test offspring for the presence of gene. Then mate heterozygous offspring to produce homozygous transgenic strains. The problems with these transgenic animals is multiple insertion, too much proteins, insertion into essential gene, lethality, insertion into a gene leading to a gene silencing, insertion into a different area can affect the gene regulation. Some of the examples of transgenic animals: superfish, increased growth and size, growth hormone gene inserted into the fertilized egg. Transgenic salmon grows about 10 to 11 times faster than normal fish. Next, glowfish. Genetically modified freshwater zebrafish, Danio rio, produced by integrating a fluorescent protein gene from a jellyfish into embryo fish. Next, transgenic mouse. Alzheimer's mouse. In the brain of Alzheimer's patient, dead nerve cells are entangled in a protein called amyloid. Mouse made by introducing amyloid precursor gene into fertilized egg of mice. Onco mouse. Mouse model to study cancer, made by inserting activated oncogenes. Smart mouse. Biological model engineered to overexpress NR2B receptor in the synaptic pathway. This makes the mice learn faster like juveniles throughout their lives. Next, transgenic pig. Enviro pig. Pigs have trouble fully digesting a compound known as phytate found in many cereal grains used to feed them. Transgenic pigs are created by introducing phytase gene of E. coli. Phytase enzyme is thus produced in the salivary glands of pig. It degrades indigestible phytate with the release of phosphate that is readily digested by pigs. Pigs for organ transplant. Pigs with human genes in order to decrease the chance of organ rejection by human body. Transgenic livestock. Bioreactors whose cells have been engineered to synthesize marketable proteins. more economical than producing desired protein in cell culture. Transgenic cattle. Transgenic cows are made to produce proteins lactoferrin and interferons in their milk. Prion-free cows resistant a mad cow disease. Transgenic sheep for good quality wool production. Transgenic goats could express tissue plasminogen activator, antithrombin 3, spider silk, etc. in milk. Next, transgenic rabbit. Alba, the EGFP, enhanced green fluorescent protein. Bunny, created in 2000 as a transgenic artwork. Transgenic monkey. Andy was first transgenic monkey born in 2000. Andy stands for inserted DNA spelled backwards. Achha. An engineered virus used to insert the harmless gene for green fluorescent protein into Andy rhesus genome. Prove that transgenic primates can be created and can express foreign gene delivered into their genome. Okay, these are transgenic animals. I think you understood very well about the transgenic animals. Thank you for listening my class.